So this is very different from the things that we have been doing so far. So we've had several talks during the summer school about label-free quantification and how that works. So you have several different runs for each of your conditions. You have a run, and using MaxQuant, you are able to get very good identification and quantification rates for the proteins across samples. So if you do MS1 labeling, for example, with SciLac, the files that we had before, well, you can have up to 3 channels in which you can combine 3 different conditions in one run, and instead of 3 runs, do only 1 and analyze everything again in MaxQuant. What MS2 labeling does, on the other hand, is allow you to even multiplex higher. And the labels are not going to be detectable in your MS1 scan. Why? Because they are isobaric. They have the same, at least nominal, identical mass. And this helps us for, as I said, multiplexing, so putting a lot of samples together. The latest and greatest is 18-plex TMT, where you can put 18 samples together. I will come to that later. And so you end up with less measurement time if measurement time is very valuable to you. And variability between runs is inherently non-existent because there is only one run. And then the way the fundamental difference is that in label-free quantification, for example, we use the MS1 signal to do the quantification, where here you would have another channel, the tags that you are going to use for quantification, which I will come to. So in short, we have peptides from different samples coming into the MS1 spectra as a single peak. We have less measurement time. We have increased precision, but not necessarily increased accuracy. And we have in the MS spectra, not just the peptide fragment ions, but also what we call the reporter ions, which are used for quantification. So to get the general idea, we start with iTRAQ, developed by SciEx. TMT, which I will explain in the next slide, is developed by Thermo Fisher. The basic ideas are the same, but let's start with the simple example of a 4-plex isobaric tag with iTRAQ. So what happens is we have, and if I can get my pointer, yes, we have such a molecule, we call it a tag, and it has 3 main parts. It has a reporter ion, which we will use for quantification. It has a balancer group, which makes sure that the tags that we have in our reagents are of the same nominal mass. And we have this amine-specific peptide reactive group, which can bind to our peptides. What's happening is that the tags have different masses. So in a 4-plex iTRAQ reagent, the smallest mass is 114.1. And this is the mass of the reporter ion. And together with the balancer group, it adds up to 145.1. The same thing happens when we have a reporter ion, which is slightly heavier than the previous one, and the balancer group is slightly lighter than the previous one. We have 4 such tags in the 4-plex, allowing us to mix 4 samples together. So we have sample 1, 2, 3, 4. We tag 1 with the first one, 2 with the second one, 3 with the third one, and so on. We mix them together. We have peptides of the same mass now. They are all tagged, hopefully, if everything works out well and you have a good efficiency in your protocol, in your labeling. And then you have these all appear in the same MS1 peak. Now, what happens is we do our MS1 scan, we choose the ions that we want to fragment, and we fragment them. What happens in an iTRAQ label is that we have two places where the fragmentation happens. The reporter ion is separated from your peptide, the tag, and the balancer group is lost as a neutral loss. So you don't see the balancer group at all anymore. You have the reporter ion in the lower mass range. So you have, in this case, you would have 4 reporter ions, 114, 115, 116, and 117. And you have your normal fragment spectra on the other side for your peptides. And it is the relative intensity differences between the reporter ions that we use for the quantification, right? I will come back to that again. So this is TMT, this is iTRAQ, and the TMT is using the same concept. There's some chemistry magic happening again. We have 3 parts, a mass reporter, mass normalizer, balancer group, however you like to call it, and the reactive group. Here you have the fragmentation happening here. So you don't lose the mass normalizer. That's the difference between iTRAQ and TMT. And the other difference is that iTRAQ has 2 versions, 4-plex and 8-plex, whereas TMT has 6, 10, 11, 16, and 18. So in iTRAQ, we had differences of one unit, but in TMT, we can have mass differences between the reporter ions as low as 6 millidaltons, which means that we need specific high-resolution machines to be able to actually differentiate these reporter ions from each other. So you need about 50,000 resolution at about 130 m/z to be able to get the 6 millidalton difference in your MSMS spectra. So this is TMT 16-plex. Again, we have the structure. You label your samples. The peptide is tagged with this label. And when you do the fragmentation, we have 16 reporters in our MSMS spectra. And these reporters look like this. So we have the normal one, which has no heavy isotopes in it, 126. Then we have 127 with heavy nitrogen. And we have 127 with heavy carbon. And this is where the 6 millidalton difference is. So between the heavy nitrogen and the heavy carbon. And the reason behind this is that the nuclear binding energies of the isotopes of every element are different. So we're taking advantage of this very, very small difference to be able to go up to 16 or 18 plex. So this is how a typical labeling experiment looks like. In this case, we only have 2 samples, so the blue and the red one. And the idea is, just to summarize again, that the lighter the reporter, the heavier the balance group, resulting in having the same nominal mass for our tags. Then we mix them after the tagging has been done, and then we introduce them to the mass spec. We get our MS1 spectra, we take our MS1 ions, and then we fragment them. The peptide is fragmented as you expect it to, and the reporter ions end up in this reporter region where we can differentiate them and use the differences in their intensities for quantification. Just another reminder, this is different from the other quantification that we do, for example, in DDA, where we use the MS1 signal, which is usually 5 to 50 times higher than the MS2 signal. But the MS2 spectra is, of course, cleaner in a sense. It has less noise in it, and we might want to use that. We might want to take advantage of that. Everything is well and good in theory, and when you design it and you say, okay, yes, I have my tags, they will be fragmented, I get my reporter ions, clean spectra, cool. But of course, we have challenges. And the idea is to overcome these challenges and do very good science. So the challenges, the factors to consider when you are deciding to do an isobaric labeling experiment or go with a label free or do something else are the following, right? So there are many papers, there's literature out there, and people like to publish such papers and people like to read such papers, and they're informative. But at the end of the day, what happens is that you are the one who should be choosing the type of experiment you want to do. So are these factors important for you? Are they controllable for your lab or not? Should you go with a label-free approach or with a labeling approach? These are the questions that is really like something that everyone should answer for themselves. So if machine time is valuable for you, then you go labeling, for example. So labeling efficiency is very important, and there, I mean, there isn't much we can do on the computational side. If the labels are not there and the signal is not there, we can't do much. So I won't be talking much about that. That's really what you guys do in the lab, and you follow your protocols and get good labeling efficiency. Then there's the isotope impurity, which I will talk about, how we deal with that. There is the co-fragmentation ratio comparison problem. Then we have dynamic range, ion saturation, and the unique and shared peptides. And then I will talk about how to compare between n-plexes. So let's say you've done one experiment with 18-plex, 16-plex, 4-plex, whatever, and you've done another one. And now you want to compare these together. So that's not easy. And we provide some tools in MaxQuant and Perseus that help with that. I'll mention them, and then Daniela will give you more details on that in her talk. So let's start with isotope impurity. So what can happen? It's chemistry, so it can sometimes be dirty somehow, where you have impurities in the labels that are being manufactured. This is expected. This can come from just the error in manufacturing or the natural isotope patterns that occur in reporter ions, similar to any other chemical structure in nature. And what can be done to tackle this problem is to have correction factors that the manufacturer usually provides with the reagents. So we can use these correction factors to get rid of these errors. It's not always perfect, but it does work. So you might sometimes get a signal in a channel that you shouldn't because of the impurities. So you might have a little bit of a signal. The correction factors would reduce these signals, but can sometimes not be able to completely get rid of them. So this is stuff that we keep in mind. And it has a significant effect on larger ratios than the smaller ones. The next challenge is the co-fragmentation, and when you have compressed ratios, what happens here is that when you have your MS1 and you have your isolation window, this happens in other experiments too, but it is a specific, like more significant problem in labeling experiments, where you are picking up a contaminating ion, which also has tags on it, which is in a different ratio to the actual peptide ion that you're interested in. So let's say you've picked up the blue ion here, the target ion, and the ratio there in your actual sample is 1:10. But then you pick up the red one as well, you take it to fragmentation, and the red reporter ions are contaminating your channels. And then the ratio is compressed. I have another slide, which I will show to you there. So these are things that are taken into, like, these are the challenges that you might face. So as I said, here, for example, we have an experiment where we have a yeast digest, and we have like different channels, so 1 to 6 to 7 to 8 to 31, and the ratios are known. So it's 10 to 4 to 1. And on the left-hand side, we have a human cell line where the ratios are 1-to-1. And we deliberately contaminate it, right? And then we can see that instead of the true 10-to-1 that you should have seen, when you have a contaminating ion, the ratio is compressed to 3-to-1. Now, maybe it's so worse that you actually don't see a difference then. It can also happen. And this is kind of arbitrary. It's not always compressing, because it can also be the case that it increases the ratio. It's just the majority of the time, because you expect the majority of the proteins to be at the same 1-to-1 ratio, the majority of the time you will have compression. And having a very good performing liquid chromatography would help with this. So you would have better separation. So the more care you put into your LC system, the less you will have problems. The narrower your isolation window, the better. And of course, if you have good software that can take care of this, you should also be okay. Another way to get rid of this problem is to go to MS3. So in this approach, what is done is that fragments from the MSMS spectra are chosen for another round of fragmentation, which would give you a different spectra. And what you have to consider here is that the first round of fragmentation, you don't want to get rid of your tags, because if you do get rid of them, you can't use them for quantification. So the tags stay there, and we select usually the most abundant ones, and then the other contaminating ions would not be there. And we do this in the MS3. The problem is not every machine can do that. So you need like tribrid instruments, which are usually a little bit more expensive. And then you're also losing a lot of machine time. So you're doing another extra scan, and it has to be high resolution, and so on and so forth. So another thing to keep in mind. There is another challenge, which is when the reporter ions are saturated. So there's a very nice publication on this matter. You can see the citation there. If you have too many ions for fragmentation, which is funnily enough the 1E6 that we usually use, the default values, is actually too much. So in this paper, they suggest that you come down to 2E5 or even 1E5 is suggested by some of the labs who are active in this field. If you have too many ions, then what can happen is that the peaks will just overlap each other, and you won't see the 6 millidalton difference between your channels. So another thing to consider when you set up your method, right? When you're setting up the method on the machine, you reduce your maximum ion target from the default to 2E5 or 1E5 or whatever works for you best. Otherwise, you might be losing some of the signals there. Okay, final thing. Now we want to compare between different n-plexes. There are these two publications which you can refer to, where we talk about the approach that we have in MaxQuant and in Perseus for comparing with confidence between n-plexes. So one thing that we do is we have an isobaric matching between runs, where similar to what happens in DDA match between runs, is when you have an identified peptide in one run, but for many reasons, an unidentified peptide in the other run, you transfer the identification. So here you can see it better. So you have this peptide here, it's been identified nicely, but in the other one, it hasn't been, and we can just transfer the identification there and get rid of a lot of missing values by doing that between the n-plexes. So another thing is that the reporter ion intensities between n-plexes can differ. And for that, you need a normalization method. And we have a specific PSM-level normalization inside MaxQuant, which allows you to actually normalize the reporter ions in a way that they are comparable to each other. And we show this very nicely here, where we don't have the normalization applied here, but we have it in the right-hand side. And you can see that we can very nicely cluster together the samples that should be clustering together. So the reporter ion intensities have come to a comparable level, so they are in the same range. And by doing that, we reduce the variance between the ratios that shouldn't be too different from each other significantly. Okay, so that's it. Thank you very much. I think we have time for a few questions.
Thanks a lot. About the intensity that you explained, the reporter ion saturation, was it about MS2 intensity or?
Yes.
Okay.
Yes. So when you have your reporter ions there, what can happen if you have too many ions going in? They will just merge because the intensity is so high.
But are you talking about fragmentation of MS2 for MS3 or?
So the way to solve that is to go to MS3 and to reduce the target ion.
Okay.
Right.
And the match between runs in your new strategy, can we also turn it off because the TMT-Plexes could be having different origins? Still, you want to compare them? Or is it by default there?
This match between runs is different from the normal match between runs.
Yeah, you said the identity, if it is there in one plex, it finds it in the other plex. So it could be that these samples are from, for example, different human people who may have really different proteomes. So they are really different from each other and you don't want to do that. So is it possible to skip it?
I think Jürgen has a comment on that.
Well, you can switch it off. You can switch on and off for sure, but on the other hand, wouldn't one better rethink the design so that one randomizes all these very different samples? I mean, that sounds like you put similar samples in one n-plex and then some samples that are similar to themselves, but not to the other ones in another n-plex. I mean, one can do, but I mean, one could also randomize all of that altogether, which might be better.
Okay, great. And the last thing, do you think the ratio compression problem is it always really like, I mean, we don't always have 10:1, right? It could be like 3:1. Do we also have that problem if the ratio is smaller?
Yes. Yeah, you will see that problem. It doesn't matter if it's 10:1 or 2:1. If you have a contaminating ion coming in, then you will also have the reporter ions there. So it will have an effect on the ratios at the end for sure.
And something that I forgot, if you use 6 plexes out of 18, for example, and you do a search on MaxQuant, do you have to change any parameters in your setting?
So if you have 6 plexes,
you have the chance to use 18 plexes, but your experiment has only 6 of them, and you want to run a search on MaxQuant. Do you need to change anything?
No, you don't have to. Then just many channels are empty. It doesn't matter.
Okay, great. Thanks a lot.
You're welcome. I have a question over there.
Thank you, Hamid. So in the last slide where you're using the word normalization, this one, is this comparing to 8 plexes or what are you? The blue and the red, are they different samples?
Blue, you mean here?
Yeah.
So the blue is when you don't use normalization,
Oh.
and the orange is where you do use it.
So you're actually normalizing for mixing, is that right?
We are normalizing so that we are able to compare between different, like let's say you have 2 8-plex experiments, and you want to compare between them. So now you have 16 samples that you're comparing.
So you're comparing different plexes.
Different plexes,
All right.
between plexes. So thank you very much.